This brief introduction to genetics will focus on Gregor Mendel and the important contributions that he has made to our understanding of genetics, but we'll also take a look at a few examples of phenomena that don't, at least at first sight, seem to align with basic Mendelian principles as examples of how our understanding of genetics has increased over the years. Let's begin by pointing out that Mendel was working with pea plants, and he noticed that these pea plants had a variety of characteristics that showed up in either or types of situations. So, for example, the seeds could be round or wrinkled. They could be yellow or green. Flower color could be purple or white. So he did various controlled pollinations, and he kept track of how these various characteristics showed up in subsequent generations, and based on his findings, he noticed that there were distinct patterns that were showing up. So as one example, let's take a look at a cross between a purple flowered plant and a white flowered plant. When he crossed these two, simply by doing controlled cross-pollinations, he would then let the seeds develop and the pollinated flowers, in other words, the peas, as we see in this figure here. He would then take those peas, plant them, and take a look at the individuals that were produced, in other words, the offspring or the first generation, often referred to as the F1 or first filial generation. Notice in this instance that the F1 generation contained all purple flowers. Somehow the white flower characteristic disappeared in that generation. But he took this one step farther and said, well, what if we were to cross-pollinate a bunch of these F1 individuals and see what happens? So he did that, and sure enough, in the next generation, referred to as the F2 generation, the white flowers reappeared. Something very interesting was going on. So this table summarizes some of his results. So for example, in the cross we've been talking about so far, crossing a purple with white flowered plants, the F1 generation, not illustrated here, recall though all of those flowers were purple. But then if you cross-pollinate the F1 individuals, he found that about 705 individuals in the F2 generation were purple flowered, whereas about 224 individuals were white flowered, a ratio of about 3.15 purples to 1 white. Notice this ratio, about 3 to 1. That same ratio was observed regardless of which characteristic he observed. Again, this is all in the F2 generations. So clearly with something going on here, very distinct pattern. Somehow there must be a way to explain these observations. So this is where the genius of Mendel really comes into play. He essentially took these results and sort of worked backwards to figure out, okay, how can we explain what's being observed here? So, in essence, he made the following conclusions. He rationalized and figured, well, again, keep in mind, this is the 1800s. This is before he knew about DNA and the structure of DNA and genes and so forth. But he basically said, well, there must be alternate versions of these things we'll call genes. Again, not really knowing what these things were, but alternate version of genes account for this variation in traits that he was observing. These different versions of genes we simply call alleles. So, for example, you might say there's a gene for flower color. One version of the gene, or one allele, codes for purple flowers. The other allele codes for white flowers. For each trait, an organism gets 2 copies of alleles. So, in other words, one from each parent. We tend to think about these alleles in terms of dominant and recessive, and fair enough, this explains Mendel's results. So we generally say that the dominant allele is expressed over the recessive allele. And this would explain in part why he was seeing this variation in flower color in terms of purple versus white flowers, just as one example. It's very common to come across Mendel's laws or principles. So let me briefly point these out as well. So if we talk about Mendel's laws, you might come across Mendel's first law of inheritance. What this refers to is the law of segregation, or the principle of segregation. In essence, all this really refers to is the fact that alleles, or essentially chromosomes, segregate or are separated. They're partitioned out in separate cells as cells undergo meiosis during the formation of reproductive cells. So again, the law of segregation simply stating that alleles segregate during the formation of sperm or eggs. You might also come across Mendel's second law, also referred to as the law of independent assortment. This essentially is illustrated in this figure here, and this explains why meiosis can provide so much genetic variation in sexually reproducing populations. So think back to these stages of meiosis. Think back to metaphase 1. During metaphase 1, if you have a diploid cell like we see here, think about how the chromosomes line up as pairs. So we could have, in this hypothetical example, during metaphase 1, we could have chromosomes lining up like we see toward the left, or perhaps these two bottom chromosomes are flip-flopped, and we could have an arrangement like we see here on the right. Whether this arrangement on the left or toward the right occurs is totally random, totally a chance event. It just depends on how these chromosomes happen to line up during metaphase 1. If you then take each of these cells and run them through meiosis, what you'll see is that you will get different combinations of alleles depending on how the chromosomes line up during metaphase 1 of meiosis. So the basic idea of independent assortment is this idea that chromosomes or the alleles on those chromosomes line up independent of each other. So the alignment of one pair of chromosomes is totally independent of the alignment of an adjacent pair of chromosomes. So let's now go through a quick example of how we can do a genetic cross using a simple human genetic trait. For example, earlobe attachment. Toward the top here in image A, we see free earlobes. Toward the bottom in image B, we see attached earlobes. So let's just use this as an example of basic Mendelian genetic cross. So it turns out that if we just look at this one trait, earlobe attachment, we can call this a monohybrid cross. Again, just referring to the fact that we're just keeping track of one trait. It turns out that if we look at free versus attached earlobes, the alleles that code for free earlobes are dominant to the alleles that code for attached earlobes, which we would then say are recessive. So let's take a look at a situation where we're going to cross two individuals, one that happens to have free earlobes, one that happens to have attached earlobes. If this is the scenario, we just said a moment ago that the alleles that code for attached earlobes are recessive. So we must know the genotype of the individual with attached earlobes. So let's say the individual with attached earlobes is a female, the free earlobed individual is a male. So the genotype of the female with the attached earlobes must be homozygous recessive. We could indicate that in a variety of ways. We typically use letters, and for recessive alleles, we typically indicate that by using lowercase letters. So the genotype of the attached earlobe person must be, let's say, just for example, lowercase f, indicating homozygous recessive. Individual with free earlobes could be homozygous dominant, capital F, or heterozygous, capital S, small f. Just for the sake of convenience, let's assume that both of these individuals in this cross are homozygous. So crossing two individuals with the genotypes shown here. The question is now, okay, if they were to have children or to have offspring, what would be the genotypes and phenotypes, or the physical appearance of those individuals? Well, the first step that you always do when conducting genetic crosses is figure out what types of gametes each individual can produce. In this example here, it's pretty simple. Regardless of how many millions of sperm the male produces, or how many thousands of eggs the females produce, they can only contribute one type of allele. So for example, the male can only contribute a dominant allele, or a capital F. The female in this example can only contribute a recessive allele, or lowercase f. If these individuals mate, they produce offspring. All of the offspring in the F1 generation would have a genotype which would include one dominant and one recessive allele. So we would refer to that as heterozygous, or containing two different versions of that particular allele. Phenotypically, all of these individuals would have free earlobes because they have at least one copy of the dominant allele. This obviously is not easily accomplished in people, but let's just say, hypothetically, if we took a bunch of F1 individuals and had those individuals mate, so a bunch of males and females from this F1 generation, have them mate, what types of offspring would we predict in that type of cross? So again, we're talking about crossing two heterozygous individuals Again, the first step is to always ask, okay, what types of gametes can be produced? Notice in this situation, each individual can contribute either a dominant or a recessive allele, so a capital or lowercase f. So given that, what then are the possible combinations of alleles that could be produced in the offspring from this particular cross? Well, a simple way to figure this out is to construct a Punnett square named after the person who first figured this out. So let's go ahead then and construct a Punnett square for this cross and see the types of offspring that are predicted in this particular genetic cross. So I've just constructed a 2 by 2 Punnett square here because each individual can produce 2 different types of gametes. So essentially all you have to do is indicate the type of gamete or alleles within the gametes along each side of your square. You can put the males down the left if you want, females across the top region. It doesn't really matter. But in this case, both males and females can produce gametes that contain either a dominant allele or a recessive allele. So we'll just write the gamete possibilities along each side there, and then you just simply bring these together and say, okay, if this sperm happens to fertilize this egg, the offspring produced would have that genotype. Likewise, if these two come together, the offspring would have that genotype, so on and so forth. And so for this particular example, we get the following scenario. We have essentially produced 4 hypothetical individuals. Of those 4 individuals, notice one of them is homozygous dominant. 2 of them are heterozygotes, and 1 is homozygous recessive. So we can talk about a phenotypic ratio. In other words, the actual phenotype or what these organisms actually look like, a phenotypic ratio of 3 to 1, meaning 3 individuals, or let's say 3 out of 4, would have free earlobes, but 1 individual would have attached earlobes. So basically 25% of all the offspring produced from these individuals would be predicted to actually have attached earlobes. The remaining portion, 75%, would be predicted to have free earlobes. We can also talk about a genotypic ratio. So for example, one of these organisms has a genotype of FF. We could say 2 of these organisms are heterozygous, both a dominant and recessive allele, and then 1 organism is homozygous recessive. So again, a genotypic ratio we might indicate by saying 1 to 2 to 1, indicating again 1 homozygous dominant to 2 heterozygotes to 1 homozygous recessive. So what we've just done is made predictions. We've said, okay, we would predict these results based on this cross. Also notice this ratio here, 3 to 1. Notice that is exactly what Mendel was seeing when he was looking at the F2 generation of his pea plants. And again, keep in mind how fundamentally important this is. He basically worked backwards. He said, okay, I'm finding these ratios that I see here, 3 to 1. How can I explain that? He basically worked backwards and figured out the whole process of meiosis and its role in sexual reproduction and a creation of genetic variation in populations. So what we just went through was, again, a monohybrid cross. We just kept track of 1 characteristic. We could do the same thing with 2 characteristics simultaneously. We would call that a dihybrid cross. The basic steps are the exact same. The first thing you do is figure out the genotypes of your parental generation, then figure out the types of gametes that each parent can produce, and then bring those gametes together in a Punnett square to find out the genotypes, or I should say the predicted genotypes, of the offspring produced from that cross. So let's now take a look at a few examples of how genetics is a little more complex than what we've just presented. For example, linked genes. Just by fortunate chance, most of the characteristics that Mendel was looking at involved genes or alleles that happen to be found on different chromosomes. It turns out if you're dealing with linked genes, we're talking about genes that are found next to each other on the same chromosome that could produce different types of ratios than what Mendel observed. And of course, if he happened to choose traits that dealt with linked genes, that might have really made it difficult for him to explain those results and come up with our basic understanding of genetics. So linked genes can confuse things a bit because, again, they don't follow typical predicted Mendelian ratios. We can also talk about this as a concept of incomplete dominance. Previously, we talked about one allele as being dominant to another allele. That's not always the case. Consider, for example, these plants that produce red flowers and white flowers. When you cross these two individuals, what we see is that the offspring are pink. So, which is dominant, the red or the white? Well, it's not entirely clear at this point. So we say that's an example of incomplete dominance. A similar phenomenon, yet subtly different, is codominance and multiple alleles. So, for example, think about the ABO blood typing system. This is a situation where we're talking about 3 different alleles, not just 2 like we've talked about previously, but 3 different alleles. One of those alleles is recessive. So if an individual is homozygous recessive for these alleles, they would have blood type O. But what about blood type A and B, or blood type AB? Well, it turns out that there are 2 other versions of the alleles that code for either the A proteins or the B proteins. Those alleles are codominant. So A isn't dominant to B, B isn't dominant to A. They are equally dominant to each other, and so that if a person has alleles for both the A and the B proteins, well, they'll both be expressed, and of course they will have blood type AB. So again, that's an example of codominance. Epistasis. This is a situation where the presence of one gene has an impact on the expression of another gene. So as one example of illustrating epistasis, let's take a look at the color of fur in Labrador retrievers. If you think about it, you can talk about yellow labs, chocolate labs, and black labs. So these different colored dogs, turns out that the fur color is due to the presence and the expression of 2 different genes. We'll call them an E gene and a B gene. So, for example, the genotype of the E gene determines whether or not the dog will produce dark pigments in their fur. So if a dog has at least one dominant allele for the E gene, indicated in the upper right here, they will produce dark fur. If they're homozygous recessive for the E gene, they will produce yellow fur. Furthermore, if we take a look at the dogs that have at least one dominant E gene, now let's take a look at this B gene. If they have at least one dominant B allele, well, they'll produce black fur. If they're homozygous recessive for the B allele, notice that they'll produce brown fur. Again, the point is whether they produce dark fur versus yellow fur, and whether or not the dark fur is black versus brown, that's all due to the interactions between these 2 different genes, again referred to as the E and B genes. So again, that's an example of epistasis. There are also a variety of quantitative traits. In other words, these are not either-or situations. For example, height. Obviously, height comes in a wide range of variation. It's not like there's either a tall person or a short person. Obviously, there's everything in between. So again, quantitative traits is difficult to think about in terms of basic Mendelian genetics. Pleiotropy. This is a situation where a single gene can have multiple effects. So, for example, if you consider the gene that codes for hemoglobin, disruption in that one gene could lead to a whole cascading series of effects. So again, pleiotropy or pleiotropic effects, these are simply situations where single genes can have many different effects. You can also think about environmental effects on phenotype. So, for example, some organisms, like the fox toward the left, their fur changes color at different times of the year, right, depending on environmental conditions. Genetics haven't changed, simply the environmental conditions have changed, resulting in different phenotypes. Toward the right, we see a cat. This is a Siamese cat. You perhaps have seen these before, but it turns out that notice the different coloration on different parts of the cat's body. So the cells they're located more toward the central portion of the cat's body. It turns out those regions are just warm enough that the enzymes involved in producing the pigment just simply don't work very well. So as a result, those portions of the cat's body tend to be light-colored, whereas the tips of the tail, tips of the ears, and outer face, and so forth, just a bit cooler, so those enzymes are functioning and producing the pigment involved. Again, keeping in mind that all the cells they're genetically identical, right? But they're the phenotype that we see depends totally on the environmental conditions. In this case, slight variations in temperature in different portions of the cat's body. The final point I'll mention very briefly is this idea of epigenetics. You're probably going to hear an awful lot about epigenetics in the near future. This is a large and rapidly growing field of study, which basically refers to this notion that yes, DNA itself and the sequence of bases in the DNA, that plays a huge role in genetics. You think about these different alleles, dominant, recessive, that's all due to differences in DNA bases and so forth. But epigenetics refers to heritable changes beyond just the sequence of bases in DNA. So we're talking about various chemical modifications of the DNA molecule itself that, again, can have a long-term imprint on the DNA. Again, I won't say much more about it at this point, but epigenetics plays a big role in human health, medicine, and a whole variety of other fairly important fields of study. So that concludes our brief discussion of genetics.